Hello everyone. Today let us study about powdery mildew disease. In my last class we had studied about downy mildew disease. Today let us study in detail about powdery mildew disease. So before going to the details of powdery mildew disease, let us study why it is called as powdery mildew. This is due to the visible symptom on the surface of the infected part of the plant. In my last video lecture, we had also studied about the difference between downy mildew and powdery mildew. In case of downy mildew, we could see the presence of the visible pathogen in the form of white cottony mass, that is, the accumulation of the sporangiophores and the sporangia at the lower surface of the leaf. That is the reason it's called as downy mildew. In case of powdery mildew, as the name suggests, it is due to the accumulation of powdery mass on the surface of the leaf. Powdery appearance is due to the accumulation of the conidiospores or ascospores which have been formed on the surface of the leaf infected part. That is the reason it's called as powdery mildew. This powdery mildew disease is caused by the organisms belonging to the family Erysiphaceae of the subdivision Ascomycotina. Another important thing which we have to note down here is these group of organism grows superficially on the surface of the host. Only it affect the epidermal tissue of the leaf has been affected in this case. Whereas in case of downy mildew, the entire tissue of the plant were affected. Here, the parasite is found to be affecting only the epidermal region of the host. If you can see the image which I have represented at the left side of this particular slide, we can see the diagrammatic representation where we could see the leaf epidermis and something called as mesophyll, where you could see the accumulation of the host of the pathogen on the surface of the host tissue, especially on the leaf epidermis. That is the important characteristic feature. You can also call it as a ectoparasite. That is, you can see this particular parasite only on the host surface. Another important thing which we have to note down here is they are obligatory parasite. They are strictly parasite. They are found growing only in the presence of the host. That is the reason they are called as obligatory parasite. However, there is a genera and a species called as Blumeria, B-L-U-M-E-R-I-A, Blumeria graminis, subspecies Hordi. Blumeria graminis, subspecies Hordi. This has been successfully grown even in laboratory condition. It can be grown as a cultures. That is important. Please make a note. This powdery mildews, most of the species of the family Erysiphaceae, they cause powdery mildew. They are strictly parasite. They are obligatory parasite. Grows only in presence of the host. But Blumeria graminis, subspecies Hordi, has been grown successfully even in culture media. That we have to remember. And this powdery mildew disease is been widely spread all over the world, and most of the plants, especially the angiosperms, gets affected. Most of the vegetable, fruit producing, as well as some of the commercially important plants, as well as the plants producing cereals, pulses, all the most of them, even ornamental plants, most of them get infected by this group of organism which causes powdery mildew, except the plants belonging to the gymnosperms. They are this pathogen cannot infect gymnosperms, maybe due to the presence of thick waxy layer, since they are xerophytes, because of the presence of thick waxy cuticle, or maybe due to the presence of resins in them. This would have made this particular pathogen not infecting them. So once again, I will repeat, this powdery mildews, or the common species, or the common group of fungi belonging to Erysiphaceae, and they cause powdery mildew disease. The symptom is the presence of powdery mass on the surface of the leaf, or any of the infected part, especially the aerial part of the plant. It can be the leaves or the twigs or the stem or the buds or the flowers or the inflorescence fruits, etc. So, and also we have to note down that this is one of a airborne disease. The propagule, infecting propagule, can be present, that can or disseminated through air, or this propagule can also be persistent even in the plant which has would have grown in the previous season. Still, if it has been found in the same region as a debris, or if the same infected plant has been cultivated again or allowed to grow again, the infection chances of infection can happen. And another important thing which we have to note down here is, unlike downy mildew require a rainy season or a monsoon season, or a water medium is required for the process of the spread of the disease. However, this powdery mildews can also be spreaded during a warm weather with some amount of humidity is enough for them, for the spread of the disease, because they doesn't require any film of water for the process of dissemination. However, at higher temperature during summer, these mycelium, or these pathogen can produce the dormant structures called as Clistrothecium. So let us study about those Clistrothecium and a little later. So and also this powdery mildews reproduces sexually by the product formation of the spores called as conidiospores. In this diagrammatic representation, you can see how the conidiospores have been produced. These conidiospores are produced from a special structures, short structures called as conidiophores. This conidiophores, they produce chain of conidiospores. These are the asexual spores which are produced by this organism during favorable condition. During unfavorable condition, that is during the summer season, they produces a special structure called as Clistrothecium. They are also called as ascocarp. These ascocarp are produced due to as a product of sexual reproduction due to the fusion of ascogonium and antheridium. They undergo a process of fusion, and as a product of which Clistrothecium are produced. Clistrothecium is a kind of ascocarp which is a ball shaped structure without any opening. So those are called as Clistrothecium. These Clistrothecium produces asci. Inside them they produces asci, wherein which asci in turn produces ascospores inside them. So this is an overview of powdery mildew disease. Then, coming to the pathogens or the casual organism which causes powdery mildew disease. Most of the species which belongs to the family Erysiphaceae, this we have to remember, belonging to the family Erysiphaceae of the order Erysiphales, of the class Plectomycetes, of subdivision Ascomycotina, they cause powdery mildew disease. Here I have listed out some of the pathogens of Erysiphaceae which causes powdery mildew disease. As I was mentioning, powdery mildew disease is being found in pulses, cereals, as well as in vegetables, flowering plants, fruits, as well as in case of cucurbits, even in forest trees, etc. So, some of the important genera which causes this powdery mildew disease goes on like this. Plumeria species, it causes powdery mildews in case of cereals and grasses. All these wheat, rice, ragi, jowar, all these plants belongs to the family Gramineae, that is grass family. They get infected by Plumeria species, which causes powdery mildew in them. Likewise, we have Erysiphaceae, which causes the diseases in case of cucurbits, crucifers, beets, tomatoes, legumes, legumes, that is all pulses. In these group, you can come across the infection with that of Erysiphaceae. And likewise, you have Levulula species, which causes powdery mildews in case of tomatoes. And you have Microsphaera species causing powdery mildews in many shade trees and woody ornamental plants. And you have Phylactinia species causing again powdery mildews in forest trees, Podospira species causing powdery mildews in case of apples, pears, etc. and Sperotheca species causing powdery mildews in strawberries, gooseberries, even in peaches, roses, etc. And We have Ancinula species, Ancinula nicata. This is one of species which causes powdery mildews in case of grape. These are some of the dominant group of powdery mildew disease causing organisms. Now, let us study about the identification characteristic feature of some of the organism of the family Erysiphaceae which causes powdery mildew. This is important for the point of your competitive examination. This we have to note on here the characteristic feature. This is mainly due to the characteristic feature or the structure of the clistothecium. Based on the structure of the clistothecium, especially based on the appendages on the clistothecium, and also based on number of asci produced inside those clistothecium, the delimitation of the genera has been done. So let us study few examples of or the key characteristic feature, how we could differentiate them. So if you observe these images which I have put it over here. So for example, in the image which I have put towards the left side, diagrammatic representation, the first clistothecium A which has been marked A is Erysiphaceae. Erysiphaceae here, which is one of the dominant group of organisms which cause disease, as I was mentioning, in case of pulses, and even in case of cucurbits you can come across the infection caused by Erysiphaceae species. So here in the clistothecium produced here is, as I was mentioning, based on the appendages. What are these appendages? First of all, the clistothecium is an ascocarp which is formed due to the as a product of the sexual reproduction. That ball-like structure which has been formed, the ascocarp, is called as clistothecium. Over the surface of the clistothecium, you can find some hyphae which has been projected outside. Those are called as the appendages. In case of Erysiphaceae, the appendage is simple. It is simple. Appendage is simple, or it can be sometime even be branched. It is simple, but the number of asci inside them is many. However, in case of B, if you come across, the appendages is like that of Erysiphaceae. That is, it is simple, but the number of asci or ascus is only one ascus can be found in the B. That is, the characteristic feature of it's a characteristic feature of Sparotheca species. Sparotheca species, In such group of organism, you can find the Clistothecium having appendage which is simple, but the number of asci, that is, we can find only one ascus inside that Clistothecium. Remember, the distinguishing characteristic feature of Erysiphaceae and Sparotheca is the appendages are simple, but in case of Erysiphaceae, you can find many asci. It can range from 3 to 6 asci can be present in case of Erysiphaceae, whereas in case of Sparotheca, the appendages are simple, but only one ascus can be found inside the Clistothecium. That we have to remember. The structure of the Erysiphaceae, the wet mount lactophenol stained image I have put over the right side of this particular slide. Please make a note. And if you see the structure C, the Clistothecium C, that is the characteristic feature of Uncinula species. Uncinula species, Uncinula nicata, as I was mentioning, the one which cause the powdery mildew in case of grapes. The distinguishing characteristic feature of this Uncinula species is the kind of appendage. If you see the appendage, it is having at its tip, you can find the structure is coiled, or it's also called as cercinate or hooked structure can be found at its terminal region. You can also see the wet mount of Uncinula species towards the right hand of this particular slide. Uncinula species, important for your competitive examination. The appendage is simple, but at its terminal region it is coiled or cercinate, or it is hooked. That's the characteristic feature of Uncinula species. Likewise, you have Phylactinia species. Phylactinia species, here the appendage is having the appendage is it looks simple, but it is straight. It is straight with bulbous base. That is the important characteristic feature. Phylactinia species, the Clistothecium is having several asci inside them, but the appendage is straight with the bulbous base. That is the characteristic feature of Phylactinia species. Likewise, you have Powdery species, where the Clistothecium the here in case E and F looks similar, isn't it? The E, Powdery species, whereas F, Microspharia species. In case of Powdery species, the Clistothecium has only one ascus. The appendage is dichotomously branched at the tip. Even in case of Microspharia species, the appendage is highly branched, dichotomously branched at its tip, but the distinguishing characteristic feature of Microspharia and the Powdery is Microspharia has many asci inside Clistothecium, whereas Powdery has only one ascus inside the Clistothecium. These are the distinguishing characteristic feature of some of the genera of the family Erysiphaceae. So once again I will repeat, Erysiphaceae and Sparotheca, they are distinguished in having the number of asci. In case of Sparotheca, you can find only one ascus, whereas in case of Erysiphaceae, you can find many ascus ranging from 2 to 6 ascus can be present inside the Clistothecium. However, the appendage is simple. In case of Uncinula species, the appendage is showing which is having the appendage tip is slightly coiled or hooked, or it is also called as cercinate. Appendage can be found in case of Uncinula species. In case of Phylactinia, you can find the Clistothecium which is having the appendage which is straight, but it has got a bulbous base. However, in case of Powdery and Microspharia, both are having the appendages which are showing dichotomous branches at its tip, In case of Podosperia, it has the Clistrothecium has only one ascus, whereas Microsperia has got many ascus inside the Clistrothecium. This is the distinguishing characteristic feature. Students, please make a note. This is very important for the point of your competitive examination. Now, let us move on to the powdery mildew disease. Let us take the example of powdery mildew of grapes. The pathogen which caused this disease is Ansinula nicata. This is a organism, as we all know, belongs to the division Eumycota, subdivision Ascomycotina, class Plectomycetes, and the order Erysiphaceae, family Erysiphaceae, and the genus which we are studying is Ansinula, the species is Nicata. This is one of a disease which has been spreaded all over the world wherever the grape has been grown. In India, it is found to be epidemic of Maharashtra, Gujarat, Tamil Nadu, and Andhra Pradesh. And even in Karnataka also you can come across this particular disease. In France in 1850 to 1855, this disease was epidemic and was responsible for the setback of wine industries. And coming to the symptoms of this disease, as I had discussed before, this is one of disease which is being spread or disseminated, or the spread of the disease or the favorable condition for the spread of the disease is cool or warm or humid area or a dry climate where no medium of water is required for the spread of the disease. That is, warm climate, cool or warm climate with some amount of humidity is required for the spread of disease. No medium of water is required for the spread of this particular disease. During summer, they produce a special structure called as Clistrothecium, during which the sporulation stops. So now, coming back to the symptom of the disease, you can come across the symptom on the aerial part of the plant, as this is one of the airborne disease where the dissemination happen through air, where the infecting pathogens or the propagules or conidiospores or ascospores or the infecting propagules which causes primary infections. These symptoms can be, as I was mentioning, can be found in any aerial part of the plant wherever the infection has happened. How the symptoms are? You can find the accumulation of powdery or dusty patches on the infected leaves or the stem or the berries or the inflorescence or the flowers, etc. This is one of the symptom, and on the fruits also you can find the accumulation of such kind of powdery mass. These powdery mass are nothing but the conidiospores which you can observe. During unfavorable condition, you could see the formation of reddish or blackish spots here and there, or the speck here and there on the surface of the leaf. This is due to the production of Clistrothecium in that particular region. So these are some of the disease symptom. Once again I will repeat symptoms, that is the accumulation of powdery mass on the surface of the infected part of the plants. Can be any aerial part of the plant. On the fruit also such accumulation can happen. Such fruits which have been found infected, they may not mature properly, they may not develop properly, they are leathery, and also the juicy content in such fruits are very less, and you can see the dusty powder, white powdery mass on the fruits also, and also you could see the rupturing of the wall of such infected fruit can also happen. So these are some of the disease symptom of powdery mildews. Now Let us study the disease cycle of Encinula nicata, how actually it causes disease in case of grapes. If the proper sanitary measures or the disease management has been not done properly, even in the plant, the infected plant which was there in the previous season, if such plant has been infected, you can see the buds. Infected plant has got some buds which would have got infected, which has got a propagule or the pathogen in the form of mycelia or a clistothecia, or the spore, either a ascospore or the conidia can be present. These fungus overwinters, and these dormant birds which are present during favorable season, they start germinating. They germinate and they they just grows into the young shoots. Once such shoot has been produced, you can see the deposition of powdery mass on these infected axillary buds which have been grown into young shoots. This is due to the formation of conidiospores from the fungus which are already present in those axillary buds. They get accumulated on such young stems, which you can find the spore formation and all. We can find the powdery mass all over those young buds. These spores which has been produced, they get disseminated through wind. We know this is one of the airborne diseases. They get disseminated through wind, and they can fall on the suitable host, that is on the grape. It falls on the suitable host, they start germinating. Once the ascospore or the conidia, if it fall on the suitable host, they start germinating. You can see the image at the right hand side of the slide. At the base you can find the stem view, where you can find the spore which has landed on the epidermis, producing germ tube, and also it producing the aprosorium, the swollen structure at the tip. It's called as aprosorium. With the help of this aprosorium, they can also penetrate inside the epidermis, producing hostorium, and at the same time they start producing mycelium also. They grow into mycelium also, which runs parallelly to that of a epidermis, we have to note down. And at the same time they produce hostorium, which are finger like projection, which has been projected only, they are confined only onto the epidermal tissue. They produces projections, finger like projections, called as hostorium. Through this hostorium they start absorbing the nutrition, and also at the same time they start producing the conidiophore. From this conidiophore, conidia, a chain of conidia are produced in basipetal succession. At that particular moment you can find the symptoms which I have put some images on the leaf as well as on the fruits. On any infected part of the plant, you could see the deposition of dusty or powdery mass, white powdery mass you can find due to the production of conidiospores and the mycelium on the infected part of the plant. These dusty or powdery mass can spread to entire leaf, even onto the lower surface of the leaf, even onto the fruits, inflorescence, flowers, etc. And this is how the symptom you can find. And at the same time, during summer season, this particular organism, they go for the process of dormancy. As a product of dormancy, you can find the development in the same dusty powdery mass on the leaf. You can find black speck like structure which would have been formed. That is nothing but the clistothecium which has been developed. Clistothecium is a ball-like structure which has been produced on the surface of the leaf. This is the product of the sexual reproduction due to the fusion of ascogonium and antheridium. You can find the process of somatogamy, which leads to the formation of dikaryotic hyphae, and which leads to the production of ball-like structure. As I had put in the image, you can find ball-like structure called as clistothecium, which can break open and release large amount of acai and ascospore. At that particular time during summer season, you can find such kind of black spot formation, that is nothing but the formation of Clistothecium. These Clistothecium, once they releases acai and the ascospores, the cycle can continue. And another important thing which we have to note down in the case of powdery mildew of grape is the Clistothecium, which produces appendages, and where these appendages are circinate or hook-like or coiled. That is also the characteristic feature of Ancinia nicata. So once again, I will repeat the disease cycle. If you can find the plantation having any chances of infection, even in axillary birds, where you can find the deposition of the propagule or the pathogen in the form of mycelia or in the form of Clistothecium, etc., they overwinters and they start developing. Once the axillary bird also develop and gives rise to the young shoot, the young shoot, before it grows, would have been completely covered over by the fungus. These fungus produces the chain of conidia. These conidia gets disseminated through wind. Once they land on the suitable host, on suitable substratum, they germinate, produces mycelium, and those mycelium can undergo process of sexual reproduction, through which the conidia formation happen. At that particular moment, you can find the accumulation of this dusty spore on the entire surface of the leaf, as well as on the fruit or flower or inflorescence peduncle, etc. And at the same time, during the summer, you can find the formation of the Clistothecium, which are the product of sexual reproduction. These Clistothecium, which in turn, during favorable condition, they breaks open, which releases large amount of acai and ascospores. And these ascospores again can infect the healthy plant, and the cycle can continue. Coming to the powdery mildew of rose, the infecting pathogen is Sperotheca panosa. Sperotheca panosa subspecies rosea is the pathogen which causes powdery mildew in case of rose. So here also, if you could find the infected plant, infected rose plant, as I was mentioning, the symptom is the accumulation of powdery mass on the surface of any infected part of the plant. So here, these the primary infection can be either through Clistothecium or through the mycelium or as a conidia which has been produced. So concentrate on the lower side of this particular slide. So here, as I was mentioning, the primary infection is through the Clistothecium. If you concentrate how actually this Clistothecium has been formed, you can find a infected leaf with black spots. During unfavorable condition, you could see the development of black spots due to the accumulation of Clistothecium. These Clistothecium overwinters, and the mycelia can also overwinter. During favorable condition, the mycelia can grow on the surface of the leaf, and they can produce a chain of conidia. These conidia can cause a primary infection to the healthy plant, or the Clistothecium which has been produced, they can also produce ascospores. The Clistothecium ruptures, the acai are projected outside. Once the ascus ruptures, ascospores are liberated. These ascospores can also infect the healthy plant. They cause primary infection. Once this conidium or ascospore fall on the suitable host, they start germinating, they produce germ tube. Concentrate on the images. They start germinating and they produces mycelium. The mycelium, they run parallel onto the surface of the host epidermis, and at the same time these mycelium produces hostorium into each epidermal cell, absorb nutrition, grows and develop, and at the same time they produces short conidiophores, and the conidia they produce in chain in basipetal succession. These at that particular moment you can find the symptom, the powdery mass, that is due to the accumulation of those conidia or conidiospores which would have accumulated all over the surface of the host tissue, infected host tissue. You can find powdery mass. That is how the symptom. You can find the infected rose leaves, buds and twigs which are showing powdery appearance. And these conidiospores which have been, or the conidia which has been produced, even they can get disseminated and they can also infect the healthy leaf or any part of the rose plant, and the cycle can continue. Coming to the control measures of powdery mildew disease, planting sites with good air, drainage and exposure to sun are helpful. That is, favorable condition has to be maintained to avoid the disease. Timely trimming and pruning of infected plant and part of the plant, removal of plant debris in planting area and burning them into ashes. Trimming, that is, frequent trimming has to be done, and pruning of the infected plant or the part of the plant has to be done, and also those part which has been infected, and also proper sanitary measures has to be followed. If such plant material, if it has been fallen on the soil, even such part of the plant debris has to be collected and they have to burn into ashes. Sulfur is most commonly applied. The sulfur dusting is sulfur is one of the fungicide which we are using especially for powdery mildew disease. This we have to remember. So what is this, how is actually this sulfur is effective? Sulfur in the form of sulfur dust or wettable powder can be applied. We can apply sulfur dust in dry areas or wettable powder in rainfall. Wherever the area is with rain, we can go for sulfur powder or sulfur dusting can be done. What is the mode of action of this sulfur? Why we have to use this sulfur? What actually the mode of action of the sulfur? This sulfur actually it interferes with the electron transport system. They interfere with the electron transport system where in the cytochrome of the fungi they reduces, the sulfur gets reduced to H2S. That we have to remember. Once again I will repeat, the sulfur they interferes with the electron transport system along with the cytochrome of the fungi, and it gets reduced into H2S, which is the toxic substance for the cellular proteins. So that is how the sulfur destroy the powdery mildews. Dusting, sulfur dusting is one of the important method of controlling the disease. Sulfur, what actually they does, interferes with that of a electron transport system, where in turn sulfur get reduced to H2S, where this H2S is found to be toxic to most of the cellular proteins. And this dusting can be done whenever the plant attains 6-inch long, just before blossoming, and after 40 to 50 days later this blossom has happened. Copper formulations and several organic fungicides like clinocap, benomil, sulfex, carathana, calyxin, topsin M can also be found to be effective against this powdery mildew. So how actually this copper formulation controls the disease? Copper ion is found to be toxic to all the cells because it reacts with sulfhydryl groups. Remember, it reacts with sulfhydryl group, that is SH group of some of the amino acids, and causes the denaturation of proteins and enzymes. That is the mode of action of this copper. Copper fungicides, the fungicide with copper formulation, they are toxic to cell where they react with SH group, that is sulfhydryl group of some of the amino acids, and causes denaturation of protein and enzyme. So this is also one of the method of controlling the powdery mildew. And also there is one more fungi called as Ampliomyces quisqualis. This is also one of a fungi which is found to be hyperparasite, which has been found to be hyperparasite on most of the powdery mildew causing organism. So if it can be a hyperparasite, what is the meaning of this hyperparasite? Parasite or the one which grows inside the host. If this fungi is a hyperparasite, that is the parasite on a parasite, so our powdery mildew disease causing organisms are the parasite. If this fungi can become a parasite on those parasite, and if it has the ability to kill those parasite, so that becomes the biological control method of controlling this powdery mildew disease. Once again I will repeat, proper sanitary measures has to be followed, and a good condition has to be maintained inside this plantation. Timely trimming and pruning of the infected part of the plant, and removal of the debris from the infected region. Use of sulfur compound, that is sulfur dusting, can be done, and copper formulations can be used. And Ampliomyces quisqualis, which is one of the hyperparasite, which can be used as a biological control to control this disease. And these are some of the reference books as well as the website. Thank you once again.